Welcome everyone. I'm excited to kick off today's presentation. Today we're diving into unveiling life's blueprint, an introduction to biomolecules. In this session, we'll explore the fundamental molecules that make up all living organisms. We'll look at what biomolecules are, why they matter, and how they contribute to life's intricate design. So let's get started and uncover the secrets behind life's building blocks. Let's take a closer look at polysaccharides, which are essential for storing energy and building structural strength in living organisms. These complex carbohydrates are made up of many sugar units linked together, providing a dense energy reserve that our bodies can access when needed. Additionally, polysaccharides like cellulose give structural support to plant cell walls, demonstrating their importance in maintaining the integrity of various organisms. Now, broadening our perspective, these molecules are part of what we call the 4 pillars of life, major classes of biomolecules that sustain life processes. Carbohydrates, in particular, serve a dual purpose. They are both a quick source of energy and a framework that supports cell structure. Understanding these molecules helps us decipher the molecular world within living tissues. When we analyze tissues at this level, we're looking at the intricate details of how these biomolecules function and interact, giving us insight into the very fabric of life. Moving forward, we'll explore how DNA and RNA act as the architects of heredity, storing and transmitting genetic information that defines every living organism. These nucleic acids are the fundamental code of life, guiding everything from growth to development. We'll also examine the sophisticated protein structures, which are truly marvels of biological architecture. Proteins are the workhorses of the cell, performing countless functions crucial for life. Finally, we'll touch on the elemental composition of these biomolecules, understanding what fundamental elements make up life itself, and how proteins, as the cellular workhorses, carry out their vital roles. All these components together form the foundation of understanding biomolecules, giving us a comprehensive view of the molecular basis of life. Understanding biomolecules is absolutely crucial because they are the foundation of how living systems function, grow, and reproduce. When we talk about these molecules, we're really delving into the chemistry that makes all biological processes possible. This journey we're on is designed to give you a deeper appreciation for that elegant chemistry. It underpins everything in biology, from how cells communicate to how organisms develop and sustain life. Recognizing the importance of biomolecules helps us see the interconnectedness of life and the molecular basis of biological phenomena. So, as we move forward, keep in mind that these tiny molecules are the key players in the grand biological orchestra. Let's take a closer look at the fundamental elements that make up life. These elements are crucial not only for energy production, but also for maintaining the structural integrity of living organisms. First, we have oxygen, or O. It's abundant in nature and often bonded to carbon. This bonding influences the shape of molecules, which is essential for their function. Oxygen is vital for respiration, helping organisms generate energy. Next is hydrogen. It's everywhere in organic molecules, forming part of water and many compounds. Its presence is fundamental for the structure and energy transfer processes in cells. Beyond oxygen and hydrogen, there are other key elements. Nitrogen, N, plays a critical role because it's a core component of proteins and nucleic acids. Think DNA and RNA. Phosphorus, P, is equally important, especially for energy transfer, like in ATP, and for the structural backbone of nucleic acids. Sulfur, S, might not get as much attention, but it's essential for stabilizing protein structures. It helps maintain the complex three-dimensional shapes that proteins need to function correctly. All these elements form the backbone of organic life, creating long chains and rings that build the complex molecules we see in living organisms. Now, when we talk about biomolecules, we're mainly referring to organic molecules, which are primarily based on carbon. While carbon is central, the real complexity comes from how these key elements, carbon, hydrogen, and oxygen, are arranged and interact. Most organic compounds are built around this trio: carbon, hydrogen, and oxygen, forming the backbone that supports the complexity and diversity of life as we know it. When living tissue is burned, the organic components combust, leaving behind an ash composed of inorganic elements like calcium, potassium, and magnesium. This process further reveals the diverse chemical makeup of life. This residual ash highlights the presence of inorganic minerals that are vital for various biological functions. Moving on, we see that the organic fraction of tissue contains larger, polymeric biomolecules with molecular weights exceeding 10,000 daltons. These include proteins, polysaccharides, which are complex carbohydrates, and nucleic acids like DNA and RNA. These are the complex structures that perform a wide range of cellular functions, from genetic information storage to enzymatic activity. Now, within these tissues, there is also a fraction called the acid-insoluble fraction. These are smaller biomolecules, usually weighing between 18 and 800 daltons. This fraction contains essential building blocks such as amino acids for proteins, simple sugars for carbohydrates, fatty acids for lipids, and nitrogenous bases for nucleic acids. Finally, there's the acid-soluble fraction, which includes even smaller molecules, often called micromolecules. These are the fundamental components, like amino acids, simple sugars, fatty acids, and nitrogenous bases, that make up the larger biomolecules. So, how do scientists study these tiny components of life? One common method involves grinding living tissue with trichloroacetic acid, or CCl3Q. This process helps us separate biomolecules into two main fractions, based on their solubility and size. By doing this, we can analyze and understand the molecular makeup of tissues, which is key to deciphering the molecular world of living organisms. Now let's take a closer look at these biomolecules and their versatile roles in life. Proteins are incredibly diverse. They act as enzymes to catalyze reactions, provide structural support in cells and tissues, and even transport vital molecules throughout the body, like hemoglobin carrying oxygen in our blood. Moving on to nucleic acids, these molecules store and transfer our genetic information. DNA holds the instructions for building and maintaining life, while RNA plays a crucial role in translating those instructions into action. Beyond these two major types, there are other essential molecules like vitamins, which are micronutrients necessary for various metabolic functions, and hormones, which serve as signaling molecules to regulate processes across the body. Both are indispensable for maintaining homeostasis, or internal balance. Water-insoluble molecules, such as lipids, are vital too. They form cell membranes, provide long-term energy storage, and insulate our bodies to preserve heat. Lipids include fats, which are our primary energy reserves and structural components. Lastly, carbohydrates, like glucose, starch, and cellulose, are fundamental as well. They serve as the main energy sources and are also key parts of structural elements in plants and other organisms. In summary, these four major classes of biomolecules, proteins, nucleic acids, lipids, and carbohydrates, are the pillars of life. Each has a unique structure and essential role, and understanding them helps us grasp the complexity of biological systems that sustain life on Earth. Proteins are arguably the most versatile biomolecules out there. They perform an astonishing array of functions in living organisms, from supporting structure to facilitating chemical reactions. Think of them as the workhorses of the cell, essential for almost every process. They are complex polymers, meaning they are made up of long chains, but these chains are built from smaller units called amino acids. These amino acids are like the building blocks, and their unique arrangements determine the protein's structure and function. Throughout this presentation, we've seen how vital molecules like proteins underpin life itself, demonstrating their incredible versatility and importance. Amino acids link together through peptide bonds, which are formed by a condensation reaction. This reaction occurs between the carboxyl group of one amino acid and the amino group of another, releasing a molecule of water in the process. These bonds create long chains called polypeptides, and these chains then fold into specific three-dimensional shapes. These shapes are crucial because they determine how the protein functions in the body. Now let's look at the structure of an individual amino acid. Each one has a central carbon atom, known as the alpha carbon. Attached to this alpha carbon are four groups: an amino group, which is minus NH2, a carboxyl group, which is minus Cu, a hydrogen atom, and a unique R group. The R group is what makes each amino acid different. It can vary widely and influences the chemical properties of that amino acid. The nature of the R group is important because it helps classify amino acids into different categories. For example, some amino acids like aspartic acid and glutamic acid have acidic R groups. Others, like lysine and arginine, have basic R groups. There are also aromatic amino acids like phenylalanine, tyrosine, and tryptophan, which contain ring structures that influence their behavior. Understanding how amino acids connect and their structures helps us see how proteins are built, starting with these fundamental building blocks and ending with the complex functional molecules that are vital for life. Let's talk about quaternary structure, which is present in proteins made up of multiple polypeptide chains or subunits. It describes how these subunits are arranged in space and how they interact with each other. These interactions are crucial because they form a functional protein complex that can perform specific biological tasks. Now, remember, each level of protein structure is vital. Even a small change at any level can significantly affect the protein's ability to carry out its function. That's why understanding these structures is so important. Moving on to secondary structure, this involves local folding patterns like alpha helices and beta sheets. These are stabilized mainly by hydrogen bonds between the backbone atoms of the polypeptide chain. These motifs are fundamental because they give the protein its initial shape and stability. Then we have tertiary structure, which refers to the overall three-dimensional shape of a single polypeptide chain. This folding is driven by interactions between the R groups, like hydrophobic interactions, ionic bonds, hydrogen bonds, and even disulfide bridges. All these forces work together to create the complex functional shape of the protein. And at the very start, we have the primary structure, the unique linear sequence of amino acids. This sequence is genetically encoded and forms the foundation for all the higher-level structures and the protein's final function. Remember, the precise three-dimensional structure of a protein is directly linked to its function. Proteins fold hierarchically, starting from a simple linear chain to complex multi-subunit assemblies, each level building on the last. Finally, when we look at all these levels together, we see the architectural marvels that are protein structures, each one intricately designed and essential for the diverse functions proteins perform in our bodies. Carbohydrates are a really diverse group of biomolecules, and they play a crucial role in living organisms. They are primarily known as the main source of energy. Think of them as the fuel that powers all biological processes. But they're not just about energy. They also serve as structural components, especially in plants and in some animals. For example, cellulose in plants provides rigidity, and chitin forms the exoskeletons of insects and crustaceans. At their core, carbohydrates are essentially sugars, simple molecules like glucose, and they can also form complex structures called polymers, which are long chains of these sugar units linked together. This dual nature of being both simple sugars and complex polymers allows carbohydrates to be incredibly versatile. Throughout this presentation, we've seen how biomolecules work together to sustain life, and carbohydrates are a perfect example of that. They're both the fuel and the framework for many organisms. Understanding their dual role helps us grasp how vital they are in both energy production and structural integrity. Now, let's explore some of the more complex forms of carbohydrates. Long chains of monosaccharide units are known as polysaccharides. These are quite important because they serve as energy storage or structural support in organisms. For example, starch is how plants store energy, while glycogen is the storage form in animals. Cellulose, on the other hand, provides structural support in plant cell walls. These long chains are essential for maintaining the balance between energy supply and structural integrity in living organisms. Understanding these complex carbohydrates helps us see how versatile and vital carbohydrates are to life. Now let's explore the plant's skeleton. Cellulose is another type of polysaccharide made up of glucose units, but it plays a very different role from starch. It forms the rigid cell walls of plants, giving them structure and support. Unlike starch, the glucose units in cellulose are linked in a way that creates long, unbranched fibers. This arrangement provides immense strength and makes plant cells sturdy. It's important to note, however, that most animals, including humans, can't digest cellulose. We lack the enzymes needed to break down these fibers, so it serves more as dietary fiber, aiding digestion. Moving on, glycogen is essentially the animal equivalent of starch. It acts as the primary storage form of glucose in animal cells, mainly in the liver and muscles. This allows animals to quickly access energy when needed. Now let's talk about starch, which is a key energy reserve for plants. Starch is a complex carbohydrate made up of long chains of glucose units, often forming a helical structure. This helical shape is quite useful because it can store iodine molecules, which gives starch a characteristic blue-black color when tested with iodine solution. That's a common way we identify starch in the lab. Finally, among these polysaccharides, starch and cellulose are quite distinct in their roles. Starch is primarily for storing energy in plants, while cellulose provides the structural integrity that keeps plants upright and strong. Both are essential, but they serve very different purposes in the plant's overall biology. Nucleic acids, specifically deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA, are truly the guardians and transporters of genetic information. They serve as the blueprint for life, carrying the instructions that determine how living organisms develop and function. These molecules are essential for dictating the synthesis of proteins, which are the building blocks of life. By controlling protein production, nucleic acids influence an organism's traits and characteristics. This connection highlights why understanding DNA and RNA is fundamental to grasping the broader themes of genetics and biology. In essence, nucleic acids form the code of life, ensuring that genetic information is accurately stored, transmitted, and expressed across generations. Now let's take a closer look at the structure of a nucleotide, which is the fundamental building block of nucleic acids like DNA and RNA. First, we have the nitrogenous base. In DNA, these are adenine, guanine, cytosine, and thymine, while in RNA, thymine is replaced by uracil. These bases are crucial because they carry the genetic information and form the code that determines how our cells function. Next, there's the phosphate group, which provides the negative charge and helps form the backbone of the nucleic acid structure. It links the sugars and bases together, giving stability and structure to the molecule. Then, we have the sugar component, which is deoxyribose in DNA and ribose in RNA. This sugar forms the core to which the other parts attach. Altogether, these components, nitrogenous base, phosphate group, and sugar, assemble into a nucleotide, which is the basic unit that makes up the larger nucleic acid molecules, essential for storing and transmitting genetic information. Now, moving on to slide 15, let's talk about RNA. RNA is typically a single-stranded molecule, which makes it quite versatile. Its sugar is ribose, unlike DNA, which contains deoxyribose. An important difference is that RNA contains uracil, or U, instead of thymine. So in RNA, adenine pairs with uracil, while guanine still pairs with cytosine. RNA plays several crucial roles in the cell. It acts as a messenger, known as mRNA, carrying genetic instructions from the DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are made. It also transfers amino acids through tRNA during protein synthesis and forms part of the ribosome itself, known as rRNA. Together, DNA and RNA orchestrate the complex process of gene expression and inheritance, ensuring that genetic information is accurately passed on and expressed. They are fundamental to the continuity of life.